All right, welcome to everyone here with us in Glasgow at the Ocean Sciences Meeting 2026 and those joining us online. This is our 3 p.m. local time press conference with Woods Hole. The preliminary results from LOC-02, a field experiment evaluating the effectiveness and impacts of ocean alkalinity enhancements. In just a minute, we're going to get started with the team and with an introduction from their press officer, Suzanne Pellison. But we wanted to let you know that if you're online and you have a question, you can put it into the Q&A box at any time, and we will take those questions once our panelists have finished their presentations that they're going to give you. If you have a technical problem, you can drop that in the chat, and we'll try to help you if we can. Please tell us your full name and your affiliation, and if you would like to ask your question live from online, or you would like us to read the question. If you're in the room, microphone's in the center of the table, just wave at us and start talking. So with that, I'm going to turn it over to Suzanne and her panelists.
Okay, great. Thank you, Liza and team. We're really excited today to have this press briefing here at OSM26. Our panelists will include Adam Sabas from the Woods Hole Oceanographic Institution, David Nicholson, also from the Woods Hole Oceanographic Institution, Rachel Davitt from Rutgers University, and Kristen Kleisner from the Environmental Defense Fund.
Right.
Adam, if you get us started, please.
Thanks, Suzanne. Yeah, thanks to Liza and the AGU team as well. Thanks everyone for being here with us today for this press briefing. I'm going to walk through some, we're all going to walk through some preliminary results from the recent field experiment that we conducted. But first, we always like to start this with just a little bit of level setting about why we're here and the role that the ocean plays in potential climate mitigation approaches. So if we can go to the next, you're on the next slide, great. So the ocean is the largest reservoir of carbon on the Earth's surface. This carbon to the tune of 38,000 gigatons or 38,000 billion tons of carbon is stored as dissolved bicarbonate or baking soda. And this is because the ocean is alkaline. It's the reason why the ocean can hold so much carbon in the first place. There's more than 40 times more carbon in the ocean than the atmosphere, and 15 to 20 times more carbon dissolved as bicarbonate than in all land plants and soils combined. So it's a massive reservoir. And we also know that carbon removal approaches are now being considered at greater and greater scales to supplement the large-scale emissions reductions that are needed to meet internationally recognized climate targets, such as the Paris Agreement or limiting warming to below 2 degrees Celsius. So you combine these two things, and the ocean is being increasingly looked at as a place where this excess carbon that we've already put in the atmosphere and are going to continue to emit even with large-scale emissions reductions, the ocean serves as a potential reservoir for this excess carbon. And there are a lot of techniques being looked at in terms of evaluating these approaches, researching their effectiveness, their impacts, and whether they can meet the challenge of scaling to climate-relevant levels. Next slide, please. Alkalinity enhancement is one of these approaches. Again, the ocean is already incredibly alkaline. It's the reason why it can hold so much carbon in the first place. And alkalinity enhancement is already a long-standing restoration tool. So it's been used successfully for decades to, say, balance the pH of rivers and lakes, to restore salmon fisheries, and to restore the pH in terms of the damages caused by acid mine drainage, et cetera. So you can see here an image of someone spraying lime in a lake, right? This is an example of alkalinity enhancement sort of in practice right now. The question is whether ocean alkalinity enhancement could work in the marine environment, not just in a closed lake, and whether it could be deployed for climate mitigation instead of, say, being deployed for acidity mitigation as its sole purpose. The other sort of caveat I'll say on this is that if you move to the next slide, there's already an active voluntary market in this space. So there are companies that are actively deploying alkalinity enhancement technologies and selling carbon credits on the voluntary marketplace. to the tune of the numbers that you can see here. So this is at, sort of thousands of tons and projected to get to the hundreds of thousands of tons in the next decade or so. And so this isn't just an academic exercise, but this is something that the private sector is also considering and moving forward with right now. Next slide, please. So just to orient you about LOC-NESS, the project, LOC-NESS stands for Locking Ocean Carbon in the Northeast Shelf and Slope. We are a research endeavor, interdisciplinary team of scientists, engineers, and communicators committed to a rigorous and transparent scientific evaluation of ocean alkalinity enhancement. And this project was designed to answer key questions about the effectiveness and potential environmental impacts of ocean alkalinity enhancement. We're not a company selling CO2 credits, and this project was really an experiment looking at some of these key scientific questions and should not be seen as a pathway to deploying alkalinity enhancement at scale. So now to get into what we did this summer. So we conducted in August of 2025 an experiment in the Gulf of Maine. This experiment was permitted by the Marine. Oh yes, next slide, please. Protected. So this permit was issued by the EPA Marine Protection Research and Sanctuaries Act. It took place off the coast of Massachusetts in the Gulf of Maine, an area known as Wilkinson Basin. We conducted an engineered dispersal of approximately 65,000 liters of sodium hydroxide and about 200 liters of rhodamine water tracer. This is a fluorescent water tracer dye that we use to track this patch of alkalinity enhanced water. In the best case scenario, this dispersal would lead to the uptake of about 50 tons of carbon dioxide from the atmosphere into seawater over the course of about a year is what our models are telling us. So if you want some more information about the EPA permit, please see this QR code that's in the corner there. The other thing that you can see in this image is the ship track. So those are the sort of black squiggles that reside within the predicted monitoring site that we had established through extensive baseline research of the area and our knowledge of currents and tides. So you can see that the experiment stayed well within our monitoring bounds during the 4 days that we were out there with ships and autonomous vehicles. Next slide, please. We conducted this experiment over the course of about 6 days. It involved 3 different vessels and 4 autonomous underwater vehicles. We had a research vessel that you can see in the foreground with 7 crew and 10 scientists on board. The dispersal vessel you can see in the background with a number of crew, engineers supplied by WHOI and some alkalinity handlers, as well as observers from the Environmental Protection Agency, protected species observers, and a commercial fisher as well. The Tioga, which you can see on the back right, also had some media observers and 2 others from Massachusetts Department of Marine Fisheries and the NOAA Northeast Fisheries Science Center. We also had a number of shoreside scientists working round the clock to pilot and manage our autonomous assets in real time throughout the monitoring period. Next slide, please. The real-time dispersal monitoring, which was part of our permit conditions, involved real-time data collection of rhodamine fluorescence. So this was our water tracer dye, pH, partial pressure of CO2, alkalinity, FV over FM, which is a measure of photosynthetic health, and oxygen concentrations. You can see this is looking over the stern of the dispersal vessel. This is the plume of alkalinity enhanced water coming out of the back, and you can see the Connecticut measuring these quantities as close as possible within safety bounds of this plume coming out. And I'm happy to say that we remain within the permit conditions throughout the entire duration of this dispersal. And the Mahoney was measuring simultaneously things like the pumping rate of material, pump volume, the vessel speed, and protected species observations, which we're also happy to say none were observed, which matches with our expectations of endangered and protected species in the region. Moving on to the results, we are able to directly quantify through shipboard, autonomous, and remote sensing observations total alkalinity budgets and carbon uptake as a result of this experiment. What you can see on the left is a satellite image collected from the PACE satellite. This was collected about 45 hours post-release. It picks up the rhodamine fluorescence. Through this shipboard autonomous data, we're actually able to convert that directly to CO2 uptake, and you can see the map of CO2 uptake in that patch at this time slot on the right. And so we can definitely say that there was additional CO2 uptake as a result of this experiment. On the next slide, we actually have a few other ways to get at this as well. So we looked at our pH data. We also looked at our partial pressure of CO2 data, and we ran an ocean model as well. And all of these results are plotted here on this right-hand chart. On the x-axis is the days since dispersal. So that's 1 day after we did the release, 2 days, 3 days, 4 days. And then this quantity on the vertical axis is essentially how efficient that carbon uptake is. So that you can think about this as the amount of carbon uptake divided by the total amount of alkalinity enhancement. You can see it increases with time throughout all of these metrics that we measured. And within error, they agree with each other. And our ocean model also agrees with these observations within error. What this suggests is that carbon uptake is indeed measurable. And the fact that we're able to directly compare ocean model results with these observations means that we have more confidence in running our models further forward in time and further constraining that CO2 uptake after the ships had to leave and go back to port. One note I will make is that we did use this rhodamine water tracer dye, and this is a really important component of the experiment and one of the reasons why we were able to do such a detailed accounting of the system throughout the monitoring period. So with that, I'll hand it over to David to talk about some of the autonomous technologies that were used.
All right, thanks, Adam. So I'm David Nicholson, also with Whole Oceanographic Institution. I'm just going to share a little more texture on some of the technology that we used in this experiment. So as Adam mentioned, we had a mixed fleet of autonomous systems that were complementing the shipboard measurements. And so shown here is representation of some of those. So we had 3 spray 2 gliders. These are our longest endurance, lowest power vehicles. One Embari Tethy's long range AUV, which is kind of an intermediate class, faster but still medium endurance vehicle, as well as a good number of surface drifters that passively sampled the surface and followed the currents. Those vehicles were equipped with a suite of sensors to measure ocean physics, and as well as critically pH and rhodamine to track both the patch and the impact of the alkalinity addition. All right, so next slide. Oh, and then the NASA PACE measurements that Adam measured also give this comprehensive view of the very surface layer. So on this slide here, this just kind of shows that when we kind of coordinate between all these observing platforms, we really got a comprehensive view of what's going on with this patch. And it's a little messy here, but it just shows all the observations over our period. The color code is by rhodamine concentration. Okay, next slide. And to give a taste of the temporal component, this quick video here showing that the gliders actually were out for a full month. So they were able to observe before the experiment started. And you'll see the experiment when you start to see the red colors there. And they were able to stay out well afterwards. And that gives us a good characterization of the background conditions. It also helped us to decide exactly what the conditions were before we were out there and where the optimal location to release the patch was. All right, next slide. So one of the big benefits of these vehicles is they give us a very high resolution view of the vertical structure. And so one of the key factors for ocean alkalinity enhancement is we want to ensure that patch stays in communication with the atmosphere and the surface layer. And so these each show different properties, but time on the x-axis and the upper 25 meters on the y-axis. There's a faint white line there that kind of indicates about where that surface layer is. And we saw the rhodamine really confined to that surface layer. And we also found it's detectable across over 4 days of the experiment. Next slide. So when we start to put these observations from different platforms together, you can see the consistency where you see this kind of upward, the up and to the right trend here is a distinct signature, both in rhodamine on the x-axis and pH in the y-axis, that is a signature of the addition from our experiment. And you see the consistency between the vehicles. And then if we go to the next slide, we're overall taking an approach where we pick a suite of different kind of technologies that really are complementary. So there's no kind of silver bullet of ocean observing in our fields. Different types of platforms have different strengths. Some are faster, some are longer endurance, some are better at sampling the surface, some can give us better depth resolution. And it's really the combination of these that give us this comprehensive picture. And if you go to the final one here, you'll just see how we're able to combine all these different observations from both the research ship, the autonomous vehicles and the drifters. And we're able to pull it all together into a really nice calibration that then gives us quantitative estimates from those remote sensing satellite images from PACE to give us a quantitative estimate and a full spatial extent of the patch. All right, so pass it on to Rachel.
Great, awesome. Thank you, David. So, hi, my name is Rachel Davitt. I'm a PhD student at Rutgers University. I'm going to give a brief overview of the preliminary results of our biological and ecosystem impacts monitoring from the field trial this summer. So, next slide, please. So I want to start with a quick overview of the marine food web. Go next. So first, we have phytoplankton, which form the base of the marine food web and produce oxygen, just like plants do on land. Then moving into zooplankton and fish larvae are primary consumers. They eat phytoplankton. And when zooplankton and phytoplankton die, their organic matter is decomposed by bacteria. So I just want to say, like, these three metrics are the main metrics that we are looking at. If you go to the next part. For our biological and ecosystem impacts, so big focus on zooplankton and the microbial community. Then if you go next, there's also moving up to smaller fish, larger fish, and top predators. And also just wanted to emphasize, if you go one more time, that by looking at phytoplankton, zooplankton, fish larvae, and bacteria, we are looking at the base of the marine food web and where there is the largest total biomass in the ocean before moving up to these higher trophic levels. So next slide, please. So yeah, going into our results on the microbial community. So the very base of the food web, from everything from the metrics that we have measured so far, we don't see any significant impact of the alkalinity patch on the microbial community. And in these graphs that I will show, I want to emphasize that the blue color is in the alkalinity patch and the red color is out of the alkalinity patch. And first looking at phytoplankton concentration by the surface, 10 meters and 25 meters, we can see no significant impact of alkalinity inside or outside the patch. I will also emphasize that the alkalinity patch only reached an extent of 10 meters of depth in this graph and throughout all the other graphs, and that here the circle is the mean and shows plus or minus standard deviation. So then moving into photosynthetic health, like Adam mentioned, this was from the surface from the ship's underway system. So water would come in and sensors on board would measure how well the phytoplankton community is performing photosynthetically. We also see no significant difference inside or outside of the patch. Then moving to dying phytoplankton cells, this was assessed using flow cytometry and a cell stain to see if there was a difference in the proportion of dying cells in and out of the patch. And again, we saw no significant difference. Moving on to biogenic silica or glass cell walls, many groups of phytoplankton and other microscopic organisms form their cell walls and shells out of biogenic silica. So this is important to measure the total amount in the water column. And again, see no significant difference inside or outside of the patch. And lastly, for the microbe community, looking at bacteria concentration, we see, again, no significant difference. Then moving up to the higher trophic level of the primary consumers, we have zooplankton and fish and lobster larvae. Again, with zooplankton, which we measured using net toes, we see no significant difference inside or outside the patch. Same thing with fish larvae. From the fish larvae that we counted during these net toes, we see no significant difference. And lastly, with the lobster larvae inside and outside the patch, there is no significant difference. It was really important for us to measure fish and lobster larvae with concerns with local fishermen. We want to make sure that if we weren't significantly impacting the fish and lobster larvae, which will grow up and support these fisheries as they mature. So throughout all these metrics that we've measured so far, we can determine that there was no significant impact of our field trial on the biological community, again, through these metrics that we measured, and we are continuing to analyze our data and collect more, continue our analysis. So move on to Kristen.
Great, thank you. Okay, so as the team has just shown, what this research is a project that involves adding an alkaline mineral to the water in an attempt to boost its ability to draw down and store carbon, which will hopefully have a positive impact on the environment, which could have impacts on marine ecosystems. So far, the results are looking promising. But these are ecosystems where people depend on for their livelihoods. And the impacts could show up in variable ways. And these are unknowns that are scary to many people. And so how the team has been addressing these concerns is to monitor the physical, the chemical, the ecological impacts of ocean alkalinity enhancement with sodium hydroxide in the open ocean. And why they're doing this research is really to understand what role, if any, ocean alkalinity enhancement has as an effective and safe mCDR approach. And these are new. This is a novel technique, and because of the novelty of this approach, there's a real need to understand, all of these, these questions, unpack these uncertainties, and to do that, really. There's a need to establish robust engagement and participation mechanisms. And so that's what I'm going to talk a little bit about, is the work that they've done and the work that EDF has done with the LOC-NESS team on community engagement. So I wanted to start off first with, next slide. Basically, a slide that came from researcher Sarah Nawaz at American University. She gave a presentation last week in a webinar, and so I'm borrowing this from her. And it's really articulates well why participation matters for mCDR research. First of all, it's the right thing to do. Communities and rights holders really deserve a voice in the decisions that affect them. And there need to be platforms for gaining knowledge and making informed decisions based on that information. The second thing is that there's evidence that shows that public input improves decision-making quality by bringing in diverse perspectives and local knowledge. And third, by engaging and having involvement on, sort of trying to level the playing field in terms of our understanding of what this project is, what it's setting out to do, and what any of the concerns may be, you can help to build legitimacy and trust. And I would caveat, and Sarah would caveat this by saying that if you're just engaging to gain acceptance, that's not advised, but really meaningful two-way engagement. So next slide. This figure, which depicts a beautiful flowing river, is emblematic of the engagement that the team did over the past 3 years and more. And it really was extensive. I think the team, the team realized that engagement was necessary at the outset because they were planning to conduct a field trial in the open ocean. Can you go back a slide?
Yeah,
Thanks.
thank you.
It's delayed. And their engagement really evolved from being more about thinking about how the results would be communicated out at the end of the project and shared out to everybody, to how they would be receiving and responding to inputs and feedback from the public as the project progressed. And engagement really did step up over the past 3 years, beginning around the public comment period of their EPA permit application. That was in 2023. Around that time, there was also a high-profile news media piece that came out that leaned into more of the controversial aspects of CDR. And so, this triggered a lot of response from the public saying, what is this? What is it? So in response, the team, held a number of public meetings and town halls. They hosted a survey that went out to the fishing community to hear their concerns and to receive input and feedback. And around that time too, EDF submitted a public comment letter to the EPA process calling for the LOC-NESS team to go further, that they were doing good work and that they should go further in terms of their outreach, particularly because this is such a new method. It's not yet proven, and so it warrants that extra public scrutiny. And so that's how we started to get involved with the LOC-NESS team, and we began to work with them to understand their approach to community engagement, what they had already done, what was working, potentially where improvements could be made, and really think holistically about how to provide lessons from the research and information from the research, to the wider community. Overall, the team conducted 55 public engagements over 3 years, and that really is, a testament to the team trying to do this outreach to the community. The focus has shifted over time to outreach and engagement with the fishing community, particularly the commercial fishing community, which were some of the most vocal public that was concerned with what was potentially happening. Next slide. So I'd say there were and there continue to be multiple goals for the engagement process, and these range from everything from education around marine CDR and OAE in particular, putting forward factual, unbiased information about mCDR and OAE, to hearing feedback about the main concerns, and then figuring out the way to best communicate back about how those concerns are being addressed by the research, protocols being put in place, and to do that in a way that people can really understand, because it is a highly technical, scientific project, and it's not easy for the public to understand all the biogeochemistry. So there are ways that we communicate in different forums and to the public. It's really important, and two-way dialogue is really critical, as is the need for information, to be communicated in these digestible ways to diverse audiences. Last slide. So the last thing I just wanted to finish with were some of the messages that resonated. I think Adam and the team would say in the earliest public presentations that the team gave, talking about rising temperatures and climate impacts and justifying using that to justify the need for mCDR. That didn't always go over so well with more polarized audiences, but instead just explaining that carbon removal technologies could be necessary to meet, or have been called for as a necessary part of the mitigation strategy. I think it just jumped ahead. Yeah, seemed to be somehow. So it was important to really address, I would say, to address the moral hazard issue head on, and what that is basically our pursuit of viable CDR options shouldn't detract or deter from our efforts to reduce emissions and decarbonize. The third point there is about, demystifying what is sodium hydroxide and alkalinity. So Adam showed a little bit about that in the beginning, about buffering, in oyster farms from acidification. It's also a chemical that's used
And then you need to choose the video. Can you choose the owl? Okay, and then make sure the audio is also on that. Okay, sorry to the online audience. We had a little disturbance in the Wi-Fi force, but we should be back online now.
How far back do you know where it?
I think go back like 1 point.
Okay, so around. Fourth point? Or last point?
Yeah, just remind them what your contextualize research in the multi-actor mCDR
Yeah,
space.
I think Adam also talked about this in the beginning as well, you know, being explicit about what the research is and is not, that it's nonprofit research, that it's not an endorsement. And yeah, putting that within the landscape, as Adam mentioned, of like around commercial deployment or the interest in that and where this research sits. And I mentioned, I think the importance of being transparent, whether you're an industry or an academic research group, being transparent about your position and your funding and engagement sources is really important. And then the last point, I think is pretty self-explanatory, but just that, you know, showing up and being willing to engage, being open, acknowledging that there's other values and viewpoints and concerns around this, but being willing to engage and address those concerns is really, really important. And I gave kudos to the team for really extensive and honest and you know, valuable, I think, participation and engagement process. We're still learning from it, and there's actually a ton of research. We'll be talking about more of it tomorrow in a session. We had a lot of interns working with us as well that did some incredible work to understand sentiment analysis and how messaging, sentiment of the public changed over time, and how tailoring messages to reach more diverse audiences really helped. So, yeah, thank you.
Okay, thank you. Sorry about the little bobble in our connection, but I think we're back up. And so we're going to go into the Q&A portion. If you're in the room, just raise your hand and please remind us who you are and who you are working with when you ask your question. That goes for our speakers too, if you want to remind everyone in the room who you are. If you're online, the Q&A box is there in the menu. You can put your name and question there. If you would like to ask your question live, just let us know and we will unmute you. So hand it over to the panel for Q&A. Any questions in the room?
Can you just explain a little bit about how this differs from some of those commercial ventures that you mentioned, like Vesto or Planetary Technologies? Is it that this, I mean, I understand that this is non-commercial, but just in terms of what's actually been done, apart from the, you know, it's research versus a commercial operation, you know, is the actual sort of bones about the same in terms of what you're putting into the ocean and what's been done?
Yeah, so Adam Spash from Woods Hole responding here. Our project differs in a few different ways. So we were using a different material. So Planetary and Vesta are using magnesium-based or an olivine-based feedstock. Primarily, we chose sodium hydroxide as our feedstock because we were very interested in how the ocean takes up carbon. And because sodium hydroxide is a liquid, it fully dissolves in seawater, or it fully mixes in seawater. We had the best chance at actually being able to keep that alkalinity at the ocean surface and give us the best shot at actually measuring the carbon uptake. We also, you know, because we're a research effort, we have a lot of resources and scientific instrumentation at our disposal. And so we really heavily monitored this project for carbon uptake, for environmental impacts. And we also have this modeling team, right? So we're able to directly compare now observations and models all from the same group. So that's another advantage, I think, of our approach. Yeah.
Great. We have a question online. Oh, go ahead.
Maybe I'll just throw into that is that this is David Nicholson, also Woods Hole, is that We also did this kind of an open ocean environment, which is unique from some of the coastal outfall work that most of the private companies have done.
All right. The question from Kate Selig with the Boston Globe. Does the research team have a preliminary estimate of how much carbon was removed during the trial?
So what we can say is that that value on that plot is around somewhere between it's a spread somewhere between sort of 4 and 20% of the total potential carbon uptake, which would translate to somewhere between 2 and just less than 10 tons of CO2 over that 4-day period. And I'll just emphasize that given our knowledge of the system and how the model is sort of agreeing in a sort of semi-quantitative sense with our observations, we will be able to extend our predictions of how that patch continued to take up carbon dioxide over a longer period of time. And again, in the best case scenario, it would have the amount of alkalinity that we added should have taken up about just under 50 tons of carbon dioxide from the atmosphere. And so as we develop our modeling, we will be able to give predictions of how that alkalinity continued to take CO2 up from the atmosphere, even after our monitoring assets left the water.
My name is Cornelia. I work for a Swiss magazine. I have a related question because there's also carbon dioxide removal going on land through direct air capture, and I don't know if it's possible. Can you somehow compare the efficiency of CDR between these two techniques?
Anyone else have an answer to that?
Yeah, I don't know if I'm not quite familiar enough with the kind of energy intensive demand for direct air capture to do a direct comparison. But
right. So that's one aspect, is the life cycle and the energy required to do direct air capture. Of course, direct air capture just captures the CO2 and you need to actually store it somewhere after that. Whereas alkalinity enhancement, the carbon is taken up by the ocean and essentially immediately locked up as bicarbonate or dissolved baking soda. And so the storage and the removal is done in one step, which is a distinction here. In terms of the efficiency, right, direct air capture, often you think of it as immediate. So you're immediately pulling CO2 out of the atmosphere per unit energy or whatever, whereas ocean alkalinity enhancement, as I was describing, is a longer term process that happens in the open marine system, right? So the other consideration is that how this evolves in time. Now, people have shown through modeling studies that given enough time, they're essentially equivalent in their effectiveness at negative emissions. How that translates to atmospheric CO2 concentrations and the effect, in the Earth system is a really interesting and active area of research right now.
Take another question online and then we'll circle back to Olive. James Dacey, freelancer, asks, it's understood that this is not a direct pathway to commercial development, but what is the biggest uncertainty in converting this signal into a robust tons of CO2 removed number? And also, how long do you expect the additional dissolved carbon to remain sequestered?
So in terms of, I'll take the last one first. Alkalinity enhancement is based on the sort of geochemical principles defined by the long-term carbon cycle. And we expect that this carbon is locked away for tens of thousands of years. It's one of the most durable forms of carbon removal. So that's one of its advantages, I would say. In terms of uncertainties, we see pretty good agreement with our model results, which is promising. There's been a lot of effort focused on developing ocean models to simulate carbon uptake into seawater. I think some of the other really interesting questions involve what is happening during the dispersal. How does alkalinity get transformed from that mineral component or that feedstock into alkalinity in seawater? These are really critical questions. They involve the immediate impacts to the marine environment, which are going to be the most acute right at the site of the intervention. And so I think there should be a lot more effort being placed on understanding what those impacts are, how they translate to overall efficiencies and the potential scaling of these approaches. And they have a lot of benefits too, because that's where we can really start to see potentially how some of these environmental impacts and impacts on marine life might start to come out.
Yeah, and I'll just kind of add to what Adam said, David Nicholson again, Woods Hole, is that a lot of the kind of proposed approaches for the monitoring, reporting, verification rely on these numerical simulations. And this is one of the few cases where we've really gone through the extensive observational characterization of what's going on, where we can really gain confidence in those models. So we need more instances of being able to demonstrate the accuracy of the models and their ability to represent these kind of find near field high resolution processes that then spread out over larger areas. So these research experiments where we can gain confidence through observation is a big step of lowering the unknowns.
Olive Heffernan, freelance journalist. I think my question is for you, Rachel. Could you just say a little bit more about the impact or potential impact of this substance on marine life? And also, just if you know from elsewhere what the limit is at which you could put this in the ocean without it having negative impacts? Like you must have had some information based on what you were willing to put into the ocean in the first place.
Of course. So what I can say to potential impacts on marine life, like I said, based on what we measured throughout the duration of the field trial, we didn't see significant impacts on the phytoplankton or zooplankton community as well as bacteria production. This is to say, we definitely do need to do more research on like longer term exposure at smaller amounts. Again, this is one field trial where we had a dispersal and then monitor it throughout the period. We also need to look at impacts to fisheries, and like one of our next steps with our research is to look at modeling in fisheries. So again, like while what we saw throughout the field trial, we didn't see any significant impacts of the alkalinity patch. That is not to say that there are no biological impacts of ocean alkalinity enhancement to be said ever. So again, like I said, there are further steps to be taken, especially looking at time-dependent and concentration-dependent exposure of alkalinity is one of our big next steps. And again, continuing to analyze the data that we have. And as to previous studies that we've done, our group at Rutgers and Adam's group and everyone at Woods Hole and many beyond have done lots of biological impact studies before, making sure to see like where the correct thresholds are. And this is all implemented into our field trial plan and making sure we weren't reaching those critical thresholds. This is also to speak to the zooplankton work. There was a lot of work done with copepods, specifically Callinis species, where we saw at the concentrations that we experienced during the field trial and above, not seeing any significant biological impacts. So there was a lot of work on that aspect before the field trial was completed.
Yeah, if I can just add something, right? We also worked in close contact with environmental protection agencies, staff scientists, and NOAA scientists, right? There was extensive consultation from NOAA as well, reviewing the permit criteria and making sure that our experiment did, had minimal impact on the marine environment. That was a big criterion for us. And so when these projects get into the water, ensuring from the get-go that you're minimizing impact is really important. And then, it was just, promising to see that our hypothesis of minimal impact was borne out by the data that we generated.
I have another couple of questions online. Michael LePage from New Scientist asked, how scalable is this? Where does the sodium hydroxide come from, for instance? What is the impact of making it? Or would something different be used at scale?
It's a really good question. It's something that we did not directly target with this research. We were really interested in, downstream, what is the potential for alkalinity enhancement to take up carbon dioxide from the atmosphere. But obviously, as these approaches continue to, get developed and diversify, it's really important to think about the energy requirement needed to produce these materials, the energy requirement to transport them and disperse them in the marine environment, the energy requirement to monitor for the carbon uptake and the environmental impacts, and take all that into account into the net effect of on carbon dioxide in the atmosphere. So this is what is called as additionality. And the idea is that you have to demonstrate that there was more carbon removed in some by doing the activity than by not doing the activity. And that's going to be a really critical area of research moving forward to make sure or to evaluate whether approaches like sodium hydroxide-based alkalinity enhancement or any of the other approaches might actually be able to scale. So the energy requirement in terms of the production is a big one that groups are actively looking into right now.
Yeah, and also to that point, I'll mention that a lot of the research we're doing was informing learning about how we can scale up the use of these autonomous systems, which have more potential to scale to larger monitoring reporting verification systems. So we're taking a lot of the learnings from this experiment to think about what a design for larger scale observing strategies would be.
Like follow-up from Michael, did you actually measure CO2 uptake directly, or are you just modeling it based on the dye distribution?
So I'll say the rhodamine dye was critical for us to understand the dilution, so how fast water was spreading and mixing with surrounding waters. So it was really a combination of that and the pH data and the partial pressure of CO2 data that does give us a direct constraint on carbon uptake through our observations. But I'll just again say that having that physical sort of passive or unbiased mixing tracer is really critical. It was really critical for us to actually be able to make those measurements directly.
Further questions in the room? There may be one more question that was hiding in the chat.
He just repeated it.
Did you just repeat it?
Yeah.
Okay, got that one.
All is happening again. I just have a question on the public engagement. Can you just speak about it in like layman's terms, in terms of what issues came up, what people were concerned about, and how you managed to alleviate those? I mean,
Well,
imagine there's a lot of fishing in that region.
Absolutely. And just to be clear, we did not conduct the public engagement.
Yeah.
The LOC-NESS team did. And so maybe I could turn it to you for some of that, but I can also give you, what we've heard and what we've looked at in the review. I mean, yeah, a lot of concern about impact on species. So I think the fisheries work is going to be really important. Concern that I think I mentioned moral hazard. And so that undertaking CDR as a deterrent from reducing emissions, like those topics came up. But it is still part, I think, of the, strategy to combat climate change. And so talking about it is absolutely necessary. I think the main things were around environmental impact, more so. And then of course, what the substances that you're putting in the water, worry that it's a chemical and what that means. But mainly, I think that all stems or can be aggregated into concern for the ecosystem, right? And would you add anything else more specific?
Yeah, one other area of concern was the pathway to commercialization and scale. That really came up. So that sort of really led us to be clear about our position as a research group. And the way that these technologies get commercialized, who gets paid for them, who sees the benefits, who sees the risks and the impacts, is really important to acknowledge and think about and explain clearly, because some of those concerns, right, as Kristen had said, it wasn't necessarily about this research project, but what people saw coming down the road. And of course, there are instances of activities that really ramped up very quickly in the marine environment that have real consequences for people's lives and livelihoods. And so, paying attention to that and the pathway that these potential technologies take to scaling, it's actually really important how we get there, not just that, if we might get there. And so I just want to bring that up again as a really important thing to consider.
I think that comes down to also the groups that were engaged and where some, that it was, I mentioned there was tribal engagement, fishing community, the fishing industry was engaged. I don't know if you mentioned that you had a member of the fishing industry on board when you did the trial to observe and watch. So trying to create those pathways too. I mean, that's engagement as well. It's like being really transparent about like what is the research and what you're doing when you're doing it. Because I don't know. I mean, nobody knows a lot about it except these guys, and now more. It's becoming more visible and more of an understanding around what is the science behind this and why conduct the science. And it doesn't mean, again, to the point about pathway to deployment, that everything has been answered, but you have to do the research to understand and make decisions based on what the findings are.
A question online from James Stacy, freelancer. You mentioned alkalinity enhancement in enclosed lakes, but is this the first such experiment in the open ocean?
As a scientist, I'm always hesitant to say we're the first. So, we're the first group to do a ship-based alkalinity enhancement experiment. There have been other trials, and even Planetaries, right, is connected to the open ocean via Halifax Harbor and olivine, placement in North Carolina, right? That's a mineral in sediments, but that alkalinity that's generated gets carried with ocean currents and tides, well beyond the site of the intervention. And so all of these approaches that are happening right now, these field trials, are sort of having those impacts are spreading beyond the site of deployment. That also means that the carbon removal, right, is happening in this really distributed way. And that's really important for us to think about as, again, for the scaling question about how we want to monitor both for the carbon removal over these really distributed large temporal and spatial scales, but also think about the monitoring approaches needed to evaluate impacts as well.
Any other questions in the room? Questions online? Last call. All right, well, I think that we've reached the end of the questions. So we'd like to thank our panelists for this presentation today. Thank you all for coming. If you have any follow-up questions, Suzanne is here to help you out connecting with the panelists. I put that information into the chat as well. And you can, of course, email us at news@agu.org for follow-up as well. Thanks again. And we'll see you next time. This is our last press briefing for this meeting, but the meeting goes on through Friday. So thanks, everyone.
Thank you.
Thank you.